This consortium is formed by outstanding groups, both from the academia and from the industry, sharing know-how, information, and co-developing technology with them is a way to accelerate our research, really. In that sense, being part of a consortium rather than working in isolation has been instrumental to actually move forward our research. My name is Eduard Batlle. I'm a researcher at the Institute for Research in Biomedicine in Barcelona, and my role at PERSIST-SEQ is I'm a principal investigator in the project, and I'm focused on characterizing drug-tolerant persister cells in colorectal cancer. PERSIST-SEQ is an important initiative because we understand very little about how cancer cells resist therapy. And so there was a project that was very central to the interest of my laboratory, of IRB Barcelona. And we thought it would be a great opportunity to join this fantastic consortium. Essentially, through PERSIST-SEQ, we believe we are actually gathering important information at the single cell level from individual cancer cells that are capable to escape the current treatments. In my particular case, in metastatic colorectal cancer, which is the cause of death for the majority of patients that develop metastases for this tumor. My laboratory undertakes sort of frontier research in this area of metastases and trying to find new treatments. And of course, in colorectal cancer, there's been very few new treatments approved over the last 30 years or so. And this is in part because all the new treatments have been tested in clinical trials in patients have failed. And we do not understand really the reason why they fail. So characterizing these mechanisms and the cells that are capable to bypass the therapy, we think will help patients. I think on the middle term, probably we will be able to find better therapies and therapies that are, let's say, effective rather than the ones that we have today that unfortunately fail in most cases. Tumors, cancers, and my particular case is metastases. They are not only formed by the cancer cells themselves, but rather these tumor cells are capable of attracting and instructing other cells from our body. And in particular, aggressive tumors like these ones are very good at calling, for instance, cell type called fibroblasts or cells of the immune system into the tumor. This is what is called the tumor microenvironment. And everything that is not a tumor cell itself, but cell from our body that is attracted and instructed by the cancer cells. And this tumor microenvironment is important for tumor progression and development. For instance, we know that cancer cells have to build new blood vessels. And so these blood vessels, of course, from our body, and they are tumor as metastasis in this case, as it expands in size, it requires to create new blood vessels. So the blood vessels is a part of the tumor microenvironment. We also know that the immune system tries to attack and eradicate the metastatic disease. And so what the tumor does is that it tells the immune cells, it sends instructions so that the cancer cells are protected from this attack. And all these different features and cell types that surround the cancer are called the tumor microenvironment. My laboratory utilizes, on one side, patient-derived organoids. So these are small structures that grow in 3 dimensions from samples that we have collected from patients. And these are kind of avatars, if you will, like mini tumors that grow in the petri dish that reproduce the features of the cancer in the patient. And of course, we cannot study anymore because, the patient has been generally operated, right? These tumors have been taken away. And then we also use mouse models. So these are animals that develop colorectal cancer and develop a disease that is similar to that of the patient. And those we use for particular in vivo studies, for instance, to study the tumor microenvironment. We have already identified how the tumors change when we give chemotherapy. Chemotherapy has an effect on the cancer cells. Clearly, in these model systems of metastases in mice or in organoids, when we treat with chemotherapy, chemotherapy kills the cancer cells, but we found that there are some cells that resist this treatment. And through the PERSIST-SEQ, we've been able to actually characterize which cell populations are resistant to treatment. And so this is information that will be released eventually for the field. So all the researchers in this area around the world have access to this information. And so they can now focus on those particular tumor cells. And then ourselves, we are now trying to develop strategies to eliminate the resistant cancer cells with the idea that we combine chemotherapy with additional therapies that target the persistent population, probably we could improve the treatment. Stromal cells and cells of the tumor microenvironment, immune cells, play a role in this process, and we're still investigating this aspect, but we believe they sustain resistance to particular therapies and then help the metastatic disease regenerate through sending signals and growth factors that enable the growth of the cancer cells. And so, we think it's crucial. We think that interfering with these particular signals that emanate from the tumor microenvironment, we probably can interfere with the capacity of the cancer cells to regrow after therapy. The development of the project will render tremendously valuable information for the cancer field as a whole, blueprint or a catalog of drug-tolerant persister cells in different tumor types. This information will be available for the community to explore mechanisms, and hopefully, the information will be used also to develop new therapies by us and other groups. And then in terms of technology, we are also developing new ways of profiling the drug-tolerant persister cells, and we are putting forward standard procedures to do so in the consortium. And so I think all these advances will also help the field now investigate additional aspects of this process of a drug-tolerant persister.